Hello students, welcome back to my channel CSIR Buddy. Today we are going to study about prokaryotic replication. So this is the part 1 of prokaryotic replication, and in this particular video we will be looking the basics of prokaryotic replication. So first before moving further, basic terms related to replication. So we should know some of the terms related to replication. First of all, what is replicon? A unit of DNA in which an individual act of replication occurs. So wherever in a DNA, this is the unit where the replication has to occur. So in this particular unit, there will be a formation of a bubble like structure. So now here the process of replication would occur. So this portion is known as replicon. Replicon may be linear or circular. So replicon could be seen in a linear DNA or a circular DNA. So basically circular DNA is seen mostly in bacterial cells. So they are talking about this circular DNA. Now the second term is monorepliconic. Bacteria replicons chromosome as well as plasmids. So what are plasmids? Plasmids are circular DNA. Circular DNA which are present in our bacteria are usually circular. So what do you mean by mono? So replicon is a unit and mono means single. So in case of bacteria, there is only a single replication site at a time which is present. So that is the meaning of monorepliconic. Now the thing to note is bacterial cells, the circular chromosome contains a single origin. As I mentioned that it consists of a single replication site, and DNA replication proceeds bidirectionally from the origin. Now the DNA replication would be bidirectional from the point of origin. So this is the meaning of bidirectional from the origin. And what do we mean by origin of replication? Origin of replication is from where the replication has started. Replication has started. So this was about monorepliconic. Again, multirepliconic is opposite to monorepliconic. In monorepliconic there was only a single origin. In multirepliconic there are multiple, we can find multiple origin, and this is an example of eukaryotes, eukaryotic organism. What are eukaryotic organism? In which the nucleus is bound by nuclear membrane very intactly. Eukaryotic cells contain multiple replication origin on the single chromosome. So in a single chromosome there are multiple replication sites. So they are having multiple origins. So this is known as multirepliconic. It has been asked in previous years that which one of the both bacteria or eukaryotic cells have mono or multirepliconic sites. So the answer will be that eukaryotic cells are multirepliconic and the prokaryotic cell or the bacterial cells are the monorepliconic. So moving further. Now prokaryotic replication, what is origin of replication? Okay, now in prokaryotic replication we have a origin of replication which is known as ORI-C, that is origin of replication. So there is a particular sequence related to that. It is a 245 base pair sequence which is present, 245 base pair sequence. So in this particular sequence there are three units which is termed as 13 mer, and 9 and 5 units, 1 2 3, which are known as 9 mer. So these are units which is present in origin of replication, that is ORI-C, and ORI-C constitute of 245 base pairs. Now so these are three units. So ORI AT rich region, because AT requires less energy to melt in comparison with GC. So we all know that in a DNA we have 4 nitrogenous base pairs. So these 4 nitrogenous base pair are ATGC. But in the case of origin of replication, this is an AT rich region. Why it is AT rich? Because ATs can easily melt. And why do we need melting? We need melting because we want a bubble to be formed, so that we can start our replication very easily. So therefore this region, that is origin of replication, is AT rich and not GC rich, because GC would require more energy to be breakdown. And why does it happen? Because A and T are in the form of double bond. They form a double bond, so the double bond is easy to break. Whereas in the case of G and C, guanine and cytosine, there are triple bonds which is very difficult to break. So therefore helps in unwinding DNA duplex. So as I said that it helps in unwinding the DNA duplex. So it becomes very easy to unwind this one duplex. Two short repeat motif, that is 13 mer and 9 mer. 9 mer and 13 mer. Origin ORI-C sequence 11 5 prime GATC 3 prime. So the sequence is like 5 prime GATC and 3 prime. Now what will happen? How the cell or how the DNA will be knowing that now it has to undergo replication? So what happens is that this adenosine gets methylated in order to inform the DNA molecule that now you have to undergo replication process. And this methylation is done by an enzyme that is known as DNA methylase. So in order to bring the replication, in both the strands this adenosine should be methylated. If only a single strand is methylated, सिंगल स्टैंड में अगर मिथाइलेशन हो भी गया है, then also it wouldn't undergo any further replication until and unless both the strands are methylated. Now coming back to this sequence. In 13 mer there are 3 units. How to remember this? That in 13 mer there are 3 and in 9 mer it is 5. So in 13 mer we can see this 3. So 13 ka 3 and 3 units are present. So we can remember this in this way. In 9 mer, opposite to 13, if this is 3, then this is 5 unit. So we can remember by this. So I hope that this one slide is clear to you all. Now moving further. Nature of prokaryotic replication. It is semi-conservative. Now what do we mean by semi-conservative? It means that the parent strand remain intact even after replication. We get one daughter strand and the another strand which we get is the parent one. So that property, this nature of prokaryotic replication is known as semi-conservative. That means that one strand will always remain the parent strand. Now it is bidirectional. Bidirectional मतलब कि दो डायरेक्शन में मूव कर रहा है. And it is discontinuous. मतलब एक strand will be continuously forming and the another strand will be discontinuously forming. So we can understand this with a small structure. So we are going to take 5 prime to 3 prime. If this one is 5 prime to 3 prime, जो भी नया strand बनेगा that would be 3 prime to 5 prime. In this case it is 3 prime to 5 prime and this one will be 5 prime to 3 prime. Now somewhere here is the origin of replication. So I will be writing it as ORIC. Now in this region, origin of replication, it must be 3 prime to 5 prime and this will be 5 prime to 3 prime. Now the thing to notice is that whenever we put a primer, it's always gonna synthesize from the 5 prime to 3 prime direction. Always it would make up the DNA replication in the direction of 5 prime to 3 prime. So in this case the primer will attach here and this must be, if this is 3 prime, this will be 5 prime to 3 prime direction. In this case the process will go on discontinuously. So the question arises that where the primer is getting attached. So primer always get attached to a 3 prime to 5 prime direction. And why it is happening? Because अगर 3 prime to 5 prime direction है, then only the primer will be able to synthesize in a 5 prime to 3 prime direction. Are you getting this point? Just look at this very carefully. If this is 3 prime, so this will gonna be, if we cut this is gonna be 5 prime. So if the primer is attaching over here, it must be 5 prime to 3 prime. As I have already mentioned that primer will always synthesize in a 5 prime to 3 prime direction. So you are getting my point. And now this is a 3 prime. So now the primer would attach here and this must be 5 prime to 3 prime. And now the process will go on of replication till here. So we get 2 strands. One is lagging strand, which is lagging in each step, and one is continuous strand, which is continuously synthesizing the DNA. In the same way, here also this must be 3 prime to 5 prime, 5 prime, 3 prime, 3 prime, 5 prime. So in this particular case also, here will be a primer which will be 5 prime to 3 prime, and it will continuously synthesize the DNA molecule. But in this one case also, the primer will attach over here and will synthesize the DNA which will be discontinuous. So primer will be 5 prime to 3 prime. So in this case we get two type of strand. One is lagging strand, and another one is a continuous strand. That will be a leading strand. So the lagging strand, we can also call it as discontinuous, and leading strand could be called as continuous. So this was all about the basics of prokaryotic replication. I have already shown you the bubble which is formed and how it is formed, how it is gonna be synthesizing the DNA strand. But there is lot to learn further, how this primer gets attached, what is the name of this primer. So all of this we will be going to study in our further videos, upcoming videos. So if you like this lecture, if you enjoyed this lecture, if you have any doubt, you can comment us in the comment section. The PDF for the same would be provided in the description box, so you can take the video PDF from the description box. And if you like this lecture, like this video, please like, share and subscribe to my channel. Thank you. Bye bye.